Assalamualaikum and good morning everyone. Today our group will be presenting about the essential macronutrients which is sulfur. This group consists of 5 members. They are Owen anak Jua, Jessica anak bujak, Jeff Paran Thomas, Bintang Ima and myself Nur Amira Natasha. First of all, I would like to present about the sulfur cycle in soil. As we know, in the soil, the sulfur element occurs in two forms, which is organic and inorganic. The sulfur also will be cycled within and between dust forms via mobilization, immobilization, mineralization, oxidation, and reduction process. Sulfur plays an important role in the soil since it is an essential nutrient that are required by all crops for optimum amino acids production. For additional, the change of sulfur to sulfuric acid in order to lowering the soil pH was done by the soil bacteria, and this will take for the elemental sulfur to change is 1 to 2 years. Next is the form of sulfur in soil mineralization and immobilization. Immobilization is the conversion of inorganic compounds to organic compounds by microorganisms or plants, by which the compounds become unavailable to crops. It is similar to the mineralization. Meanwhile, the mineralization is the decomposition of chemical compound in organic matter, where the nutrients in those compounds are released in soluble inorganic forms that may be available to plants. It is the opposite of immobilization. The product of mineralization of organic sulfur are hydrogen sulfide, elemental sulfur, and sulfide minerals.
Sulfur volatilization is the process by which sulfur is released into the atmosphere as a gas. This can occur through natural processes such as the breakdown of organic matters in soil or through human activities such as the burning of fossil fuel or the use of sulfur-containing pesticides. Sulfur volatilization is of concern because sulfur gas can contribute to air pollution, including the formation of acid rain. In addition, sulfur volatilization can lead to the loss of sulfur from soil, which can have negative impact on plant growth and soil fertility. To reduce the amount of sulfur volatilization, farmers can use sulfur-efficient fertilizer and practice other sulfur management strategies.
The sources of sulfur for crop production are mostly in atmosphere as a sulfur dioxide and irrigation water, which is the sulfate. It also from the organic compound, which are the animals and municipal waste. For the sources from inorganic compound, also from inorganic fertilizer, which are the ammonium sulfate, potassium sulfate, the calcium thiosulfate, and also the urea sulfuric acid. For the sulfur in plant, it could occur in two measures of amino acids, which are cysteine and methionine. The cysteine as the first carbon or nitrogen reduced sulfur products from the sulfate assimilation. It will serve the sulfur donor for made the methionine, the glutathione, vitamins, the cofactors, and sulfur compounds, which play a major role in the growth and development of the plant cell. The both are essential for the primary and the secondary metabolism of the plants. This also occurs in a myriad of fundamental processes from the photosynthesis to carbon and nitrogen metabolism. The both also occur in proteins, which is the former playing wide range of functions in proteins catalysis. This sulfur atom in the proteins forms part of a redox buffer as for a glutathione through specific detoxification or protection mechanism.
In plants, sulfur is typically found in the form of sulfate ions. Sulfate ions are molecules that consist of a sulfur atom bound to 4 oxygen atoms with a charge of -2. Sulfate ions are usually derived from sulfuric acid or sulfur dioxide, and they play a number of important roles in plant metabolism, including the synthesis of amino acid, proteins, and other biomolecules. In addition to sulfate ions, plants can also contain other forms of sulfur, such as reduced sulfur compounds, such as thiols and thiodos.
Sulfur transport in plant. Our understanding of the mechanism of sulfur and phosphate transport in plant has improved significantly in recent years through the application of molecular technique in nutritional physiology. Gene encoding transporter involved in the uptake of sulfur by plant roots and the distribution of these anions throughout the plant have been identified. Expressions of gene encoding this transporter in root is transcriptionally regulated by feedback signal that respond to plant and sulfur status. However, there is evidence that genes encoding high affinity sulfur transporter may also be regulated by nutrient requirement and the availability of purchaser required for sulfur assimilations. Gene encoding high affinity sulfur transporters are expressed in cell in close contact with the soil solutions, especially epidermal cells with associated from hair. Gene encoding other low affinity transporters are expressed in cell associations with the vasculature, where they play a role in the internal redistributions of sulfur function in plant. Sulfur serves many functions in plant, used in the formations of amino acid, protein, and oils, necessary for the formations of chlorophyll, promote the formation of mass in legumes, aids in the development and activations of certain enzyme and vitamin. It is two structural component of the 21 protein building amino acid.
Next, I will explain on sulfur deficiency symptom in plant. Whenever the sulfur status of growing plant drop below the critical level required, visual symptom of sulfur deficiency start appearing on the plant. The appearance of such symptom indicate a serious condition because crop yield can decrease even without the appearance of such symptom. Sulfur deficiency symptoms in many ways resemble those of nitrogen deficiency symptom, in which the leaf become pale yellow or light green. Unlike nitrogen, sulfur deficiency symptoms appear first on the younger leaf and persist even after nitrogen application. In cotton, tobacco, and citrus, some of the older leaves are affected first. For example, sulfur deficiency symptom on oil palm. Seedlings have small pale green to almost white frond and show some intravenous streaking, while older leaves develop necrotic spot or dead tissue followed by terminal necrosis. As for sulfur deficiency symptom on paddy, the leaf sheath and the leaf blade become yellowish. Moreover, the whole plant may look chlorotic at the tillering stage. Plant height and number of tillers are reduced. Panicles are fewer, shorter, and bear fewer grains than normal plant.